Good afternoon, everyone, and thank you for joining us. I'm Mindy Buchanan, Director of Programs at the Myotonic Dystrophy Foundation. Next slide, please. And it is my pleasure to welcome you to our special presentation of MDF's Meet the DM Drug Developers with Dyne Therapeutics. Next slide, please. First, I want to share a little bit about the Myotonic Dystrophy Foundation. We envision a world with treatments and cure for myotonic dystrophy. And the mission of the Myotonic Dystrophy Foundation is community care and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. And we accelerate research toward treatments and a cure. Next slide, please. Some of the great resources you can find on our website include toolkits and publications, including our anesthesia guidelines and clinical guidelines to share with your providers, support programs, including our support groups, and you can also find a whole calendar of events with all of our upcoming activities. You can also check out our Digital Academy, which has recordings of past sessions from both Meet the Drug Developers and education sessions, as well as all of our past conference sessions. Next slide, please. So we also have some additional upcoming Meet the DM Drug Developer sessions. On July 7th, we have AMO Pharma, and August 4th, we have Avidity Biosciences. And of course, we have 2 sessions scheduled for our drug developer sessions at the annual conference in September in Washington, D.C., which is both a hybrid, both a virtual event and an in-person event. Next slide, please. So if you'd like to know more about joining our conference, again, it's in Washington, D.C. It includes an advocacy day on Capitol Hill on the 7th, and it will be September 8th and 9th for the general conference program, and you can register on our website and learn more there. Next slide, please. All right, I am thrilled to share. We have Dr. Ashish Dugar here from Dyne. Dr. Dugar is the Chief Medical Affairs Officer at Dyne, and he has over 20 years of broad experience across medical affairs, clinical and commercial development, and real-world evidence generation. Prior to joining Dyne, Dr. Dugar served as Vice President and Global Head of Medical Affairs at Head Therapeutics, Incorporated, where he built the company's medical affairs organization. Previously, Dr. Dugar led all commercial and market access efforts for Intracellular Therapies, Incorporated, as Vice President and Head of Commercial Development. Dr. Dugar has also held executive management roles at Roche and Pfizer. He began his career at the National Institutes of Health and holds a PhD in pharmacology from Penn State College of Medicine. Next slide, please. Also with us today is Molly White. Molly is VP of Global Head of Patient Advocacy and KOL Engagement. Molly brings extensive experience in patient advocacy and community engagement to her role at Dyne, where she leads global patient advocacy and KOL engagement across the company's disease focuses. She previously served as Chief Executive Officer of the Myotonic Dystrophy Foundation, a global patient advocacy organization. In that role, she held strategic direction and administrative oversight for the comprehensive programmatic and fundraising efforts of the foundation and its staff. Molly has over 25 years of leadership level experience building, implementing, and running award-winning domestic and international community programs for organizations in both the private and nonprofit sectors. She received her MA from the University of Iowa. Next slide, please. All right, so just before we start, a bit of housekeeping here. You can definitely ask questions throughout the entire webinar. If you're using your desktop, you open the tab to the questions tab, and you can type your question right in there and click send. And if you're on a smartphone, click the question mark icon at the top of the screen, where you can also click type your questions in and click send. Next slide, please. And please consider supporting the MDF mission. If today's program speaks to you, please consider showing your appreciation with a donation to MDF, and together we can change the future of myotonic dystrophy. It's now my pleasure to welcome Dr. Dugar and Molly for their presentation. Welcome to Dyne Therapeutics. Thank you so much.
Wonderful. Thanks, Mindy. Hopefully you all can hear me. So I'm Ashish Dugar. I'm the Chief Medical Affairs Officer at Dyne Therapeutics. And as Mindy said, joining me is Molly White, someone well known to this community, and also Dyne's Global Head of Patient Advocacy. First, I want to thank Tanya Clede, Mindy, and the entire MDF team for inviting us to present at this webinar to update the DM community on exciting and recent preclinical data demonstrating that our FORCE platform, which is the basis for all of our investigational medicines, can deliver antisense oligonucleotides that target genetic disorders to the brain. As always, it's a true privilege to speak with the myotonic dystrophy community. These are our forward-looking statements. Please refer to our website if you are interested in more details. I like to begin with this slide in every presentation. At Dyne, it all starts with our mission. We're laser focused on delivering life-transforming therapies for people living with serious muscle diseases, with myotonic dystrophy type 1 being a priority. Our mission is core to who we are and what we do every day. Our focus today is on sharing some recently presented data in mice and monkeys, where we showed that we can deliver antisense oligonucleotides to the brain. I'll also remind you at the end of the presentation that we have an ongoing trial in adult onset DM1. I know that I don't have to tell you that about the disorder. You're living with it each and every day, or your family members. Very briefly, DM1 is an autosomal dominant progressive multisystem degenerative neuromuscular disease, most commonly diagnosed in adulthood. But symptoms can appear at any time from birth to advanced ages, depending on the type of myotonic dystrophy. Some of the symptoms can include the muscle myotonia, which is the inability of muscles to relax after using them, often described as muscle stiffening, followed by weakness and atrophy of skeletal muscles. Other symptoms include cardiac conduction abnormalities and respiratory distress, GI and metabolic disturbances, fatigue and sleep issues, and all of these greatly impact quality of life. And we also recognize that for a rare disease, DM1 is not so rare and often impacts multiple generations within a family. So this is a schematic that describes the path from the mutation in the DMPK gene to the clinical presentation of symptoms of DM1 that I just described. If we start with the left-hand side, basically the key point here is that DM1 is caused by an expansion of CTG repeats in the DMPK gene. Specifically, people with DM1 have abnormally high repeats of CTG, which are the building blocks of DNA, and these high repeats are seen in both blood and tissue. Now, these repeats translate as CUG repeats in the DMPK RNA. And these expansion repeats in the RNA lead to formation of large structures that capture muscle-blind-like protein, or MBNL, shown in the second chevron here, and it inhibits something called splicing, which is needed for appropriate gene regulation. So these clumps that form, that you can see these little hairpin loops, these clumps that are forming are considered accumulation of foci and occur in the nucleus. And this can be detected in both mouse models as well as humans. And these trapped proteins or clumps are needed to process RNA in the nucleus so that they can do that job of cutting RNA called splicing. And the formation of these clumps or foci is directly related to the spliciopathy, which is essentially a widespread splicing defect that leads to the clinical presentation seen in DM1. And it's this presentation it's this splicing defect that we are truly trying to address with therapeutics. And it's crucial for an ideal drug for DM1 to address the skeletal muscle issues, the cardiac issues, the respiratory issues, smooth muscle issues, as well as the CNS pathology. So currently there are no disease-modifying treatments available, limiting treatment to the management of symptoms only. So to treat the genetic basis of disease, the key is to get the therapeutic agent into the nucleus of the cells, knock down the toxic DMPK, which can then correct the splicing defect that I talked about earlier, which may lead to slowing or stopping the progression of disease. And it's Dyne's targeted delivery approach that we have built this company on to effectively and efficiently deliver our therapeutic agent to muscles and patients so that we can make a meaningful difference in the lives of patients and their families. So as I go through some of our preclinical data in a few minutes, how our platform works will become more clear. Thanks. So here I'll take a couple of minutes to describe the FORCE platform. The FORCE platform drives all of our efforts to develop and deliver muscle-targeted therapeutics. It's expressly designed to address one major issue in delivering antisense oligonucleotides to where we want them to go, which is efficiently and reliably delivering these with on-target effects and limited off-target effects. So what I mean by that is being able to target the genetic basis of diseases so that we can alleviate what the pathology is in those key tissues, like skeletal muscle, cardiac muscle, and smooth muscle. As you can see, Dyne's FORCE platform consists of 3 parts: an antigen binding fragment, or what we call a Fab, to enable targeted delivery of the ASO to muscle. On the bottom, in the burnt orange string, a clinically validated linker, and on the bottom here, an antisense oligonucleotide selected to target the genetic basis of disease. I'll start by describing these 3 components, and then I'll come back to the importance of targeting the transferrin receptor 1, or the TFR1, to drive that targeted delivery of our investigational therapeutics. So we chose a portion of a monoclonal antibody. Here it's the Y-shaped figure. So we chose the top portion of that monoclonal antibody, again, an antibody fragment or a Fab. And we believe this Fab versus a monoclonal antibody offers powerful advantages over a larger size protein because it's easier to penetrate the muscle cells. There's a lower overall protein load and potential for enhanced tolerability profile. Again, the goal is ultimately to enable targeted, effective delivery of therapeutic molecules to the tissues where they need to go. We selected the Val-Cit linker for our platform based on its stability and that it's clinically validated. That is, it's used in products that are on the market today. So we believe this further de-risks our FORCE platform. So we're utilizing the same Fab and linker across all our programs. The third component is the ASO chosen to target the genetic basis of DM1. So the versatility and modularity of our scientific platform provides flexibility to deploy different types of antisense oligonucleotides or other agents that are rationally selected to match the target biology of the disease. In our case for DM1, because the toxicity resides in the nucleus, we use an antisense oligonucleotide. If the toxicity resides in the cytoplasm, for example, we can use something like an siRNA. So this is a very modular, versatile platform that allows us to really drive our drug development efforts efficiently. So the mechanism of FORCE, as I mentioned, is different from other approaches. We're essentially using the cell's natural biology to deliver our conjugate to muscle cells. Our Fab binds very tightly to something called the TFR1 or transferrin 1 receptor. Now, these TFR1 receptors are highly expressed across the surface of muscle cells, including skeletal, cardiac, and smooth muscles. So we're able to harness what already exists on the surface of muscle cells to deliver our therapeutic with this receptor-mediated mechanism. And by targeting TFR1, we should have the potential to be able to have a wide therapeutic index and durability of effect, which should result in less frequent dosing with chronic administration. So this slide is something I've presented in the past, and this is just to show what kind of data we have generated in monkeys to confirm the ability of FORCE to get into key muscles in a large animal. Specifically, we see that FORCE leads to robust wild-type DMPK knockdown across skeletal muscle, cardiac muscle, and smooth muscles. We know there is significant distribution of the ASO into this larger species, which is really important as we think about how we can apply this platform into the clinic. Please note that these are healthy monkeys, so we cannot actually look at toxic DMPK knockdown. This is wild-type DMPK knockdown. So as mentioned, we wanted to explore the ability of our platform to cross the blood-brain barrier and get an antisense oligonucleotide into the brain. The left-hand part of the slide shows how highly vascularized the brain is. That is, there's a tremendous system of blood flow in the brain, and it's this extensive capillary network that provides the potential to enable widespread delivery of molecules to deep inside the brain tissue. Now, as a protection to the brain, there's something called the blood-brain barrier, or BBB, to prevent the passage of large molecules from the bloodstream into the brain parenchyma. On the right side of the slide, you can see that the TfR1 receptors are located on the blood-brain barrier. So this provides an opportunity to use the TfR1 mediated transport to potentially take molecules across the blood-brain barrier. In fact, other companies have shown, as well as academic institutions, have shown that using TfR1 mediated transport, or TfR1 transcytosis, they can get molecules across the blood-brain barrier. And given the ability of force to bind to TfR1, as we've shown for quite some time in skeletal, cardiac, and smooth muscle, the logical question was to ask whether force can deliver to brain with a similar mechanism. So I'll take a little bit of time to go through this, and hopefully it's a clear explanation. Next question we want to explore was how the force conjugate delivered to the brain compared to administering a naked antisense oligo intravenously, or IV, or intrathecally, or IT, versus the force conjugate administered intravenously. So we looked at ASO delivery to regions of the brain and wanted to look at sort of these sub-tissue level of cortex, caudate, putamen, amongst other brain regions. So we created a probe against our antisense oligonucleotide. And on this slide, we look at representative images of monkeys infused with force, which is the rightmost panel. And then we look at representative images of monkeys infused with unconjugated ASO, delivered IV, which is the left panel, or IT, which is the center panel. And the ASO delivery is identified by the white staining. So on the left, you can see virtually no delivery of the ASO, naked ASO, delivered IV. In the middle panel, where we have naked ASO delivered IT, you can see staining on the superficial layers of the cortex, but level of distribution decreases in deep brain to a point where it's barely detectable. So you can see the decrease over, within the brain as you go deeper into the brain. And in the rightmost panel with force delivered IV, we see widespread distribution in the cortex and deep brain regions like the caudate and putamen. This clearly shows force achieves widespread delivery of an antisense oligo. In this case, the ASO is designed to target the DMPK in the brain of monkeys. I do want to point out the importance of the HTFR DMS XL mouse model, because the next few slides provide data in this particular mouse model. So as I mentioned, the literature indicates that the toxic human DMPK RNA, that is the genetic basis of DM1, resides in the nucleus. So we developed the HTFR1 DMS XL mouse model shown on this slide by crossing mice that have the human TfR1 receptor and DMS XL mice that carry the human toxic DMPK transgene with about 1,000 or so repeats. So what I'm saying is this model expresses both the human transferrin receptor and the human toxic DMPK. And we can actually look at what happens to that toxic DMPK that resides in the nucleus, as well as look at what happens to those clumps or trap proteins or the foci that I was discussing earlier. So this animal model has the potential to give us good translatability in terms of what may happen in patients who also accumulate toxic nuclear DMPK and have splicing abnormalities. I've also presented this data to you previously, so just a reminder of what happens when we apply force in this mouse model in the heart. So when we evaluated force in the heart of this mouse model, after just 2 doses of 10 milligrams per kilogram, we observed a reduction in foci and toxic DMPK knockdown at about 49%. And again, this resulted in a very compelling splicing correction result after about 4 weeks. Now, we know that the cardiac muscle has been difficult to access in muscle diseases, and this is critical to improving the condition of patients. So we were very pleased to see this level of cardiac penetration and pharmacodynamic effect, or DMPK knockdown and splicing correction and foci reduction. We have seen similar results across other key muscles involved in DM1, including skeletal muscle, respiratory muscle as well. Now, we're really excited by the data from numerous studies in the HTFR DMSXL mouse, as we believe that this work sets a new standard for evaluating DMPK knockdown and splicing in DM1. So on this slide, we show that the force conjugate delivers to brain cortex and achieves toxic human nuclear DMPK knockdown and foci reduction in this mouse model. From left to right, we show data on ASO delivery, DMPK knockdown, and DMPK foci reduction in the cortex. On the left, we see how force leads to much greater delivery compared to controls. And this correlates to the DMPK knockdown in the cortex seen in the middle panel. And more importantly, you can see how mice treated with vehicle versus force. We see the profound reduction of the trap proteins or the foci reduction in this particular mouse model in this experiment. We showed that with force, there's about a 65% reduction of foci in the nucleus. On this slide, we show a very similar pattern and similar data in the cerebellum with force in terms of ASO delivery versus controls, significant DMPK knockdown versus controls, and then significant foci reduction, about 60% foci reduction with force versus vehicle. So altogether, these data show clearly that force can achieve delivering antisense oligonucleotides across the blood-brain barrier and have pharmacodynamic or effects that we just talked about in terms of the knockdown and the foci reduction in this mouse DM1 model. So we're really excited that overall the force platform demonstrates robust delivery of oligonucleotides to skeletal, cardiac, smooth muscle, and CNS by leveraging this TfR1 mediated mechanism. Importantly, we showed superior and widespread delivery compared to naked or unconjugated ASO administered either IV or IT. And in the DM1 model, we showed that the force conjugate achieved significant DMPK knockdown and foci reduction in the brain. We believe that the force platform has the potential to overcome that one big issue that I mentioned earlier, which is to be able to deliver oligonucleotides to where they need to go, whether it's skeletal muscle, cardiac muscle, respiratory muscle, smooth muscle, as well as the brain. And we believe this is critically important to treat the spectrum of DM1 patients. On a separate note, I want to remind you that we have a phase 1/2 study underway in adult onset DM1, and I just wanted to provide a very quick review of that study design and the assessments that we're doing. So our clinical trial for Dyne 101, called Achieve, is a global trial consisting of essentially 4 periods. We have a 6-week screening period, followed by a 24-week placebo-controlled multiple ascending dose period, where individuals receive either placebo or Dyne 101 administered every 4 or 8 weeks. This is followed by a 24-week treatment period where all participants receive Dyne 101 administered either every 4 or 8 weeks. And finally, a 96-week long-term extension period. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. We plan to evaluate the following outcomes in the Achieve trial: safety and tolerability, of course, and that is first and foremost. This is a phase 1/2 safety study, and we have patient safety first and foremost in our minds when we're conducting our program. We're also going to be looking at splicing, as well as things like pharmacokinetics, which is how Dyne 101 moves into the body, through the body, out of the body, as well as pharmacodynamics, such as looking at splicing and DMPK knockdown. We'll also be looking at several measures of muscle function and strength to help us understand how the body is functioning and changes and how the body is functioning with treatment. And we also carefully assess respiratory and heart function. With the help of our expert advisors, we're really confident that our study design is robust, is measuring key outcomes, and through all of the feedback from the advocacy community, that we are striving to manage the burden on families as we conduct the study. We're really excited to say that we'll be communicating some initial safety, tolerability, and splicing data in the second half of this year, and I will come back to this community to talk about that when we're ready. So here you can see some of our inclusion and exclusion criteria that must be met for an individual to participate in our study. For more information on study design, as well as the IE criteria and open trial sites, please visit our ct.gov posting, and you can find all of that information there. So we couldn't do our work without your support, encouragement, and sacrifice. We understand that work from countless dedicated scientists, physicians, physical therapists, and other healthcare providers across the world is needed because this disease is so devastating with no approved treatment options for people such as Suzette and Billy Dean pictured here. And then the amount of support that the community has provided is overwhelming, and we could not do any of this without you. Thank you very much for your time and attention. And if you go to the next slide. So we'll go through questions and answers that the community has provided, and then we can open it up to other questions that are coming in through the chat line. We'll be happy to take any of those questions. All right. So Molly, I will just start with the Q&A.
Sure. So the first question we got before the webinar launched was, what are the side effects of this drug or treatment? And I'm assuming by that they mean the Achieve Dyne 101 study.
Yes. I think we had many questions. We had a few questions related to the animal data in CNS. We'll get to those. We had a set of questions related to the clinical trial, and we'll go through those as well. So happy to, I think we have time to go through almost all of the questions. So to your question on the side effects of the drug or treatment. So first of all, as I mentioned, we have an ongoing trial in adult onset DM1 with Dyne 101. And so we are collecting all of that data to truly understand what the safety and tolerability profile looks like for the drug. And so that's just being collected on an ongoing basis. What we can say is that Dyne 101 had a favorable safety profile in a 13-week GLP tox study in monkeys, which is a prerequisite before we go into the clinic. And we've also, we saw no changes in terms of kidney, liver, weight, ophthalmic changes, neurologic changes, respiratory changes, cardiac changes. And so we're really pleased with the profile of what we saw in this 13-week GLP tox study.
Excellent. Here's a question. How many drugs are in the pipeline for DM at the moment?
Great. So in general, there's a robust pipeline. There's a number of preclinical programs, and then more than 3 clinical programs using a variety of disease-modifying strategies. So that's sort of in the universe of what's happening in the myotonic.
Look, there's a question around whether this is directly translatable to DM2. Don't know if you guys can hear Ash.
I am back with you, sorry.
Okay.
Yeah.
The question is whether this is directly translatable to DM2, and I'm not sure whether they meant the Achieve study or the CNS delivery in general.
All right, let me try to answer both. So first of all, as I'm sure you all know, DM2 is a CCTG repeat, as opposed to DM1, which are repeats of CTG. So we are looking at, our current therapeutic agent that is in the clinic is targeted for DM1, and so it would not be applicable for DM2. We would need a different approach to target DM2. We do recognize there's an urgent need for new therapies for DM1 and DM2, and so we are constantly evaluating our pipeline for opportunities to help the community.
There's a question around how a person can get involved in drug trials.
So in terms of getting involved in drug trials in general, as well as, this applies to Achieve too. First of all, talk about participation in trials with your physician. Get in contact with physicians that are working on clinical trials like the Achieve clinical trial. Visit the Achieve study page at clinicaltrials.gov, or for any of the ongoing trials, look at the ct.gov pages for various sponsors, because there's a ton of information in there. But really, driving this conversation through your physician with yourself and your family is probably the first step.
What is the timeline for an effective treatment to be approved for use? And will this be available in this person's lifetime?
So that's a great question. I think we're all hopeful that the timelines for drug development, approval, and reimbursement get shorter and shorter, but it is a very, very long process. Frankly speaking, development of the drugs, approval, reimbursement can take 5 to 10 years. Achieve, our trial, is designed to be registrational, and we continuously work with global regulatory authorities during the development of the drug to make sure that we're meeting the regulatory requirements, and then also working with health authorities around the world so that we can gain market authorization and reimbursement. So we're working on that path, and again, it's something we can come back to this community with when we're ready.
Just in general, what is the purpose of this new drug? What's the treatment, the physiological effects?
So maybe I'll answer it on two levels. So if we just talk about force, the platform itself is designed to overcome the challenges of delivering ASOs to muscle tissue by leveraging the transferrin 1 receptor, or the TFR1. Our overall goal is to develop therapies that stop or reverse disease progression. And as I've shown here today and previously, the force platform enables delivery to various key muscles like skeletal, smooth, and cardiac muscle, as well as has been shown to cross the blood-brain barrier. So we're really excited about the potential of using TFR1 as a delivery mechanism. In terms of the purpose of Dyne 101, the compound we are studying right now, we are looking to see how it affects adult onset myotonic dystrophy on various levels: splicing, myotonia, measures of muscle strength and function, as well as quality of life. And so that study is ongoing, and as I mentioned before, we'll have some initial data in terms of safety, tolerability, and splicing in the second half of this year.
How much will this potential therapy cost, and is insurance going to cover it?
So right now we're in early stages of the drug development process. That said, we know how important access and affordability is, and we keep these issues topmost in our minds as we move forward.
So the question here on what CNS changes happened in the animal models, and I know you talked about this a little bit, but maybe you can go into a little more detail.
Yeah, sure. So maybe the key takeaways here are that we have been able to show that using the TFR receptor, we can deliver to the brain, we can cross the blood-brain barrier, we can deliver to the cortex and deep brain regions like the caudate and putamen. And we're really excited that we're able to show this reduction, significant reduction in the clumped or trapped proteins or these foci, which is really important to potentially alleviate the the pathology associated with DM1. So that's probably the key point. Importantly, we did compare distribution of the ASO with our force conjugate delivered IV versus an unconjugated ASO delivered IV or intrathecally. So again, we are really excited that we can see such a difference in terms of delivery of the ASO.
When will this clinical trial come to the U.S.?
So Achieve, which is again the Dyne 101 investigational, the study looking at Dyne 101. It's a global trial, and we've activated sites in multiple countries. The U.S. is clearly an important community and geography for Dyne. So we're going to be generating data from our initial cohorts outside of the U.S., bring that back to the FDA and have a discussion with them about the path forward to open up sites in the U.S.
Can you speak to any congenital applications of any of this work?
Yeah, so I think right now it's early, and so I think we really need to focus on evaluating Dyne 101 in the ongoing Achieve trial, understand how it acts, and what kind of initial safety and tolerability profile develops as we dose more patients. And so this is something that we'll need to come back to the community with as we gain a better understanding of Dyne 101.
Would you say that that Achieve is designed to be preventative or intended to cure?
So again, the Force platform overall is designed to overcome that challenge of delivering ASOs to key tissues by leveraging TFR1. Our goal is to develop therapeutics that stop or reverse disease progression, and so that we are going to learn more about what our platform can do as we're in the clinic and with other work ongoing within the company. So this is really designed for now to be a chronic treatment that is tunable, titratable, and hopefully has a wide therapeutic index.
Can you talk about how the Dyne 101 is potentially going to improve heart function, particularly low ejection fraction and AFib?
Yeah, no, it's a great question. So as I probably mentioned earlier, the Achieve study is a phase 1/2 study where we are really focused on safety and tolerability as well as measuring other outcomes. Again, we have patient safety first and foremost in our minds. And so in doing that kind of study, we want to be very thoughtful about the inclusion-exclusion criteria. So we do have inclusion-exclusion criteria related to the level of cardiac function that patients may have. So right now we want to make sure that we're able to generate data in the specific patient population that we've outlined and get some initial safety and tolerability data. And over time, we would love to explore the effects on things like ejection fraction, arrhythmias, AFib, et cetera. So again, that's something that we have top of mind and we'll be looking to explore over time.
Yeah, and to your point, we get significant delivery to the heart.
Yeah, great point. Yeah, as we showed the preclinical data earlier in this presentation, we're really excited about being able to affect quite a change in the HTFR1 DMS XL mouse in terms of the cardiac manifestation.
On that same vein, can you talk a little bit about how Dyne 101 might improve lung function and particularly diaphragm performance?
Yeah, sure. So within Achieve, for example, we are looking at pulmonary function. So we do have measures of pulmonary function. So we'll be over time generating data to understand what's happening in terms of lung function. Again, we can point to some of our preclinical data that has been shown in the past on diaphragm in terms of DMPK knockdown, foci reduction, as well as correction of splicing. again, we're really excited about what we're seeing in the mouse model, and we'll see what happens in terms of lung function in the clinic as we continue with our study.
And then there's another question, and then I've got a list here from today's discussion. There's a question around, can I participate in the study? I don't have any visible symptoms.
So what I would say is, it's something that you should definitely speak to your physician about in terms of potentially participating in our clinical trial. And of course, our ct.gov website also has the parameters that are needed to be met to be able to enter into our trial. I would always advocate first, please go talk to your physician as you're thinking about participating in this trial or any trial.
So we've got some additional questions from today. Can you just quickly define again the difference between IT and IB?
Sure. So intrathecal administration, in our case, is essentially administering it into the spinal cord, and then intravenous is into a vein. So it's systemic delivery.
And maybe you can summarize again what FORCE is exactly.
So FORCE is, again, the platform upon which we have built our investigational medicines. And there's three components to it. One is a piece of an antibody or an antigen binding fragment that we've engineered to bind super tightly to the transferrin receptor 1. And again, the reason for that is because of the TFR1 receptors found on so many key tissues and muscles. We then have a linker, a valcit linker, and this linker is used to connect the Fab to the antisense oligonucleotide that we've designed to target the genetic basis of any, of a disorder. So, for example, whether it's DM1, an ASO designed to target the DMPK gene. We have a Duchenne program ongoing. So we have an antisense oligonucleotide designed to attack the genetic basis of Duchenne for 51 skip amenable for specific mutation in those patients. And then, so that linker is validated. It's used in products that are actually on the market right now. So it's really well understood, helps to de-risk our platform. And then we have, as I mentioned, the ASO that's connected to the Fab via the linker to target the genetic basis of disease. In the case of DM1, it's an ASO because ASOs are preferentially trafficked or preferentially go into the nucleus. And so that is where we want to attack the DMPK.
There's a question, are these single injections or infusions? And I'm not sure if that's related to the CNS work or Dyne 101.
So in the animal data that I presented today with the FORCE conjugate, that is administered intravenously. And if they're talking about the trial for the ACHIEVE study, we are administering Dyne 101 intravenously.
There's a question, will you do pulmonary function testing to see how the drug affects lung function during the clinical trial?
We are, yes.
Quite succinct, thank you. What is the time frame for FDA approval and being available to the market for Dyne 101?
Yeah, very, very important question. So again, right now we're in the early stages of being in the clinic, and so as I mentioned, we're really happy that we'll have some initial data to communicate in the second half of this year. And as our programs progress, I'll continually give updates to the community. We don't typically, don't typically talk about kind of where we're at from a regulatory or access perspective until we have some firm information. What I will say, I will add, we have had conversations with regulatory authorities around the world, and they've been very positive interactions, as evidenced by the fact that we have a number of clinical trial sites open, activated. patients are dosing all over the world. And so we've had great interactions. Global regulatory authorities understand what we're planning to do and are supportive. And again, as we have more information, we'll come back to the community. But it's been really positive so far.
So I'm going to jump around just a little bit, but I'll make sure we get all of them. There's a question here specifically around when is it going to be available in the US?
So again, what we're going to do is generate some initial data outside of the US and bring it back to the FDA and have a conversation with them about opening up sites in the US. As we have more clarity and firm plans around that, I'll come back to the community to talk about it.
So there's a question here. Since since force crosses the BBB or the blood-brain barrier, does Dyne 101 also cross the blood-brain barrier?
Yeah, it's a great question. So again, right now we're just doing some early exploration in our non-clinical models, and so we have the data that we've put in front of you all today, just to show that using the force platform, we can cross the blood-brain barrier. So that's our first step. And as we progress in our exploration of what our compounds can do, we'll come back and communicate this to you.
I think there's a related question here, which I think you're going to answer the same way, which is basically, since it crosses the blood-brain barrier, will this help with brain fog and depression?
So again, we're in it's the same answer. You're right. It's we're in early stages to understand things like distribution and foci reduction in the animal models. We're pleased to say that in those experiments, the conjugate was well tolerated in those animal experiments where we explored CNS effects. And so we'll just continue working on this, and we hope to be able to again address all of the symptoms and all of the different clinical manifestations of DM1 as we progress our programs.
Will your drug be administered via infusion to a vein or into the spinal cord?
So again, for the ACHIEVE study, Dyne 101 is administered into the vein. It's administered intravenously.
A very nice thank you from someone. Will you wait for current trials to get over with, or will you approach the FDA with preliminary data?
So so we want to engage the FDA as soon as possible, and we want to generate some data from outside of the U.S. where the trial is currently running, and then come back and have a conversation with the FDA. We're really excited outside of the U.S. We're able to start at pharmacologically active doses, and again, great support from a number of ex-U.S. Regulatory authorities. So we're really excited about generating some data at pharmacologically active doses, and then coming back to the FDA to talk about it.
So I think this is with regard to the CNS presentation. So can the Fab conjugates deliver ASO using endogenous levels of TFR1 without transgenic overexpression?
So it's a really good question. I probably need to dig into that a little bit more. I don't know that the mouse model we have has overexpression of TFR1. So I'll have to go back and come back to the community after talking to some of our scientists about that specific question. I don't want to give you an answer that's incomplete or wrong.
Well, you may feel the same way about this one then. So since the ASO is not allele specific, what might be the consequences of knocking down DMPK protein expression from the unaffected allele? This kinase may have a function.
Yeah, it's an interesting. We've had that question before, and so what I point to is we can go through the literature, but I point to our data. We have looked at knockdown of DMPK, wild type DMPK in monkeys, as I showed earlier. We've also done safety and tolerability. We've also done the GLP toxicology study in monkeys. And so we aren't seeing any untoward effects or consequences from a tolerability point of view by knocking down, the wild type DNPK. And so we feel very comfortable with our force conjugate. We feel very comfortable with how we've applied it in terms of Dyne 101 in the preclinical space, which has given us confidence to move into the clinic as we have. It's a great question. We get that a lot.
And I think I have covered all of the additional questions that came in during the webinar. The MDF team can correct me if I missed something here, but I think we got it.
Great.
Oh, wait, hang on, late breaking. Yes, there's a point, someone's pointing out here that DNPK may have brain function in learning and memory.
Yeah, well, so, it's possible. I think maybe I'll extend that a little bit. So why is it important to be looking at areas like cortex and caudate, putamen, et cetera? Well, the caudate and putamen, for example, are involved in learning and motor control. They're critical for various functions such as planning, the execution of movement, learning, memory, reward, motivation, emotion and social interactions. And then dysfunction in these areas could be a factor in DM1 to your point, and many other disorders, because they're strongly linked to problems with motor function. And Huntington's is an interesting example. Huntington's disease is characterized by rapid and spasmodic involuntary movements, and it's associated with the degeneration and death of neurons in the caudate and putamen. So these are really important areas to the point that the person made. And the cerebellum controls voluntary movements such as walking, posture, balance, coordination, eye movements, and speech. So it's really critical to be able to look at what's happening in these kinds of regions of the brain.
So I think that's it. Unless anyone else has questions they'd like to drop in, I think we have covered everything.
Wonderful. Well, thank you. Thank you again for your time and attention. Molly and I are incredibly appreciative of the great questions, and to MDF for giving us some time today.
Wonderful. Thank you, Dr. Dugar, and thank you, Molly, so much for joining us today, and thank you to our audience for joining us as well. This webinar will be available in our digital academy in a few days, so make sure to check back there, and I hope everyone has an amazing afternoon and rest of their week.
Bye-bye.
Bye-bye.
Thank you. Bye.